two phases of photosynthesis or two steps in which photosynthesis takes place. One is light reaction, other is dark reaction. Light reaction is light dependent because the solar energy is utilized and is converted into ATP and a reducing power is synthesized. Now, in dark reaction, using those two things, that is ATP and NADPH2, carbon dioxide will be reduced. So, basically in dark reaction, carbon dioxide reduction takes place. This dark reaction is also known as Kelvin cycle, named after the scientist Melvin Kelvin, who actually gave us all the steps in which this cycle takes place. So, how this, which product is formed, what are the intermediate products. So, complete path of this cycle was given to us by Melvin Kelvin, and that is why this is known as Kelvin cycle. Kelvin used chlorella plant. So, this experiment, Kelvin's experiment, which gave us the complete cycle, was by using chlorella, which is a unicellular green alga. He used this chlorella and gave this chlorella carbon dioxide which had radioactive carbon. So, carbon dioxide which was used contained carbon 14. So, CO2 was actually C14, radioactive. Now, when chlorella performed photosynthesis, it started using this carbon dioxide. So, this carbon dioxide got incorporated. Carbon fixation is what is going to take place. So, this carbon got fixed and he killed this chlorella after regular intervals. He took a beaker, chlorella in it, exposed it to this carbon dioxide, radio labeled carbon containing carbon dioxide, and once the reaction started, that is exposure of light and carbon dioxide, this photosynthetic alga would have started performing photosynthesis. After every few seconds, some extract was taken out, killed chlorella by, he took out this extract and killed them by dipping it in hot methanol. He killed chlorella by dipping it in hot methanol after every few seconds, maybe 5 seconds, then 10, then 15 and so on. And then he analyzed this carbon, radioactive carbon. He used chromatography also, and to trace this carbon, he was trying to see where this carbon went, and this carbon 14 was traced. When he started tracing it, it was traced in a particular form of a compound. So, then he said, okay, this carbon dioxide which is taken by the green plant gets converted into this. Then after few seconds, when few more chlorella cells were taken, he analyzed this same way, he found another compound. So, this was compound 1, compound 2, compound 3 and so on. And this is how he gave us the complete cycle, that what happens to this carbon dioxide which is fixed by the green plants. So, this Kelvin cycle is not a very simple cycle. So, what we are going to do first is we will take a simplified version of a Kelvin cycle, and then after that we will see exactly how it happens. But to understand, we need to take the simpler version of Kelvin cycle. If we divide this Kelvin cycle, it gets completed in 3 steps. We will write those 3 steps. First step is called carboxylation. Carboxylation is taking in of carbon dioxide or binding of carbon dioxide with something. So, this carbon dioxide is now utilized. there has to be a molecule which is going to receive or accept this carbon dioxide. That molecule is RuBP, rubulose biphosphate. So again, let us write down these abbreviations here. RuBP. It is a 5 carbon compound. 5 carbon compound. It is called rubulose biphosphate. Earlier it was called RuDP, diphosphate, but this is the newer version or newer name given to it. So this carbon dioxide is accepted by RuBP. An enzyme is required for this. The enzyme is called RuBIS, Rubisco. RuBP carboxylase. It is called RuBP carboxylase. Actually, this Rubisco is a very important enzyme for many reasons. One reason is it is the most abundant protein or enzyme in the living world. And in chloroplast, whatever proteins they have, this makes 16% of the total proteins. And it is found in all green cells. Every time photosynthesis takes place, it is there to help in this first reaction. Another important thing about it is that it can act as carboxylase also and oxygenase also, but that behavior changes under certain conditions. We will take those conditions later on. Normally, when temperature is normal, carbon dioxide concentration, everything is normal, at that time it acts as carboxylase. So this Rubisco helps in addition of carbon dioxide to RuBP. This first step is carboxylation. In this case, when Kelvin was analyzing everything, the first stable compound which they found was a 3 carbon compound. But scientists believe, Kelvin also and the scientists who followed after him, who did the same kind of experiments, they say that there is a 6 carbon compound which is formed. It is a very short lived compound. As soon as it is formed, it dissociates. So we will write a 6 carbon compound which is temporary or short lived. So it is just one thing which is there, but the first stable compound is a 3 carbon compound. Now to understand this cycle, we will take a fixed number of carbon dioxides. We will start with 6 carbon dioxides. So how many RuBPs are required to take those 6? 6 RuBPs are required. 1 RuBP will accept 1 carbon dioxide. Let us also keep a track of carbon numbers. How many carbons are here? 1 carbon dioxide contains 6 molecules of it. So there are 6 carbons here. RuBP, rubulose biphosphate, is a 5 carbon compound. So how many molecules? 6. So 5 6 are 30 carbons are here. And we get a short-lived temporary complex which is a 6 carbon compound, and this will immediately dissociate into a 3 carbon compound. Total number is 36 carbons, and this compound which is formed, though it is a short-lived temporary complex, it is a 6 carbon compound. So how many molecules of these 6 carbon compounds should be formed? 6 molecules. So we get 36 carbons. We have to keep a track of these carbons. Then only we will be able to understand where how many carbon compounds are going. So a small stage or a step in which a short-lived temporary complex is formed, it immediately dissociates into a 3-carbon compound. This 3-carbon compound is PGA. PGA stands for phosphoglycerate or phosphoglyceric acid. Phosphoglycerate or phosphoglyceric acid. It is a 3-carbon compound. Phosphoglyceric acid is a 3-carbon compound. So, how many molecules are formed here? 12 molecules. So, 3-carbon compound, 12 molecules of it, 12 threes are 36 carbons. So far everything is matching, the carbon numbers and all. Now, this phosphoglyceric acid undergoes phosphorylation. That means one more phosphorus or phosphate will get added. The molecule which is going to give or supply phosphorus is ATP. Each molecule, phosphoglyceric acid, this glyceric acid already has one phosphorus. One more phosphorus will get added to it. So, how many ATPs should dissociate? There are 12 PGAs. So, 12 ATPs, each one will give one phosphorus to one acid, and we will get ADP 12 and inorganic in this case only ADP will be given out. So, 12 molecules of ATP will dissociate to give 12 ADP, and this PGA will change into 1,3-di PGA. Why 1,3? Because earlier there was only one phosphorus on one carbon. Now there are two phosphates, one on the first carbon and other on the third carbon. It's just a 3-carbon compound. So 1 for first phosphorus position, 3 for the second phosphorus position. So it becomes 1,3-diphosphoglyceric acid. How many molecules? Same number. So here also 12 molecules of this. Though it is a diphosphate, but it is still glyceric acid. So it has same 3 carbon atoms. So 3 into 12, that is again 36. 36 carbons are there. The enzyme which is going to help here is a phosphorylase. So the enzyme that helps would be a phosphorylase. It can also be called a triose phosphorylase because both are 3-carbon compounds. Now, this phosphoglycerate is reduced. For this, some agent which is going to act as a reducing agent is required. In light reaction, there are two things which are obtained. One is ATP and other is this reducing power. We said both the things which are synthesized in light reaction will be utilized in dark reaction. So here NADPH2, again we can write it as NADPH plus H plus. So NADPH2 would get oxidized into NADP and it will reduce the phosphoglyceric acid to phosphoglyceraldehyde. So the next substance which is formed is phosphoglyceraldehyde. So PGAL stands for phosphoglyceraldehyde. Again, this phosphoglyceraldehyde is a 3-carbon compound. So 12 molecules of 3-carbon compounds. the carbon numbers are still 36. 12 molecules, each molecule is a 3 carbon compound. So 12 threes are 36 carbons. In this case, each molecule is containing only one phosphorus. So it is simple phosphoglyceraldehyde, and it is coming from a diphosphor substance. So this phosphoglycerate or phosphoglyceric acid had 2 phosphates, whereas when it changes from acid to aldehyde, this molecule has only 1 phosphorus. So some phosphates are given out in the form of inorganic. Let us put the numbers. To reduce 1 PGA, that is phosphoglyceric acid, 1 NADPH2 will be required. There are 12 molecules. So 12 NADPH2 will reduce 12 diphosphoglyceric acids, 12 NADP will be obtained, and 12 inorganic phosphates will be released. Now we have an aldehyde, phosphoglyceraldehyde. The first reaction, that is addition of carbon dioxide, was called carboxylation. In this part, what is happening is reduction. So the step number 2 can be termed as reduction. And if you are able to recall what happens in glycolysis, in glycolysis, phosphoglyceraldehyde changes into phosphoglyceric acid. So it is exactly reverse of glycolysis. So here it can also be written as reversal of glycolysis. But the step is known as reduction, where phosphoglyceric acid is reduced to phosphoglyceraldehyde. Now these 12 molecules, they will separate into 2 groups, 1 and 2. 2 molecules will come out of this cycle. So here there are PGAL, phosphoglyceraldehyde, 2 molecules. So how many carbons? Each is a 3 carbon compound, 2 such molecules are coming out. So 6 carbons are getting separated, and these 6 carbons will actually give us glucose. So what is being done so far? The carbon which was taken in the form of carbon dioxide has been converted into a carbohydrate, and that is why photosynthesis is also known as carbon fixation. We are fixing that carbon dioxide into an organic compound. So out of 12 molecules of PGAL, 2 have separated out of the cycle. That means 6 carbons. How many still remain? 30. So the other group is again of PGAL. How many molecules are here? 10 molecules. Each PGAL is a 3 carbon compound, 10 such molecules. So how many carbons are here? 30 carbons. So out of 36, 12 molecules, they separate into 2 groups. One is of 2 molecules and other is of 10. Or you can say 6 carbons would separate here and 30 would again get separated. Now these 2 molecules which are separated, they would help in glucose synthesis. These 10 molecules of PGAL, they would rearrange themselves to form RUBP. So basically these 10 molecules are helping in regeneration of RUBP. So step number 3 of this cycle is regeneration of RUBP. From a 3 carbon compound, we will make a 5 carbon compound here. Plus, this is phosphoglycerate, or sorry, phosphoglyceraldehyde. That means each molecule has how many? Only 1 phosphorus. And here, each molecule has 2 phosphates, that is biphosphate. So now these 10 PGALs will change into RUBP by rearrangement of carbons, plus phosphorus is also needed, that will come from ATP, and this ATP will break into ADP, phosphorus, and energy will be utilized. So we have 30 carbons here in the form of 10 molecules of 3 carbon compound. They will rearrange to give us 6 molecules of 5 carbon compound. Total has to be 30. So now how many ATPs are required here? 6 ATPs, because 1 ATP will give 1 phosphorus to 1 RUBP. Ultimately there are 6 RUBPs, so we need 6 phosphates. So 6 ATPs, and it will break into 6 ADPs. So we have divided this complete cycle in 3 steps. We means the scientists have divided, and this is how we study it. So now the carbon numbers are proper. Reason why we took it 6, because we wanted to show 1 glucose synthesized in one go. If we start with 1, this will come in fractions. So then it becomes a little complicated to understand. How many ATPs are required to synthesize 1 glucose? 12 ATPs here and 6 here. So the total, like we need to see how expensive it is to synthesize 1 glucose. So for 1 glucose molecule synthesized, we need 18 ATP. 18 ATP are required. And how many NADPH2? In the entire cycle there is only one place where we need it. So 12 NADPH2. So the ATPs which are synthesized during light reaction and the reducing power, that is NADPH2, which is synthesized during light reaction, both get consumed in dark reaction. So these are the 3 things which we are consuming: ATP at 2 places and NADPH2. Okay, let us quickly go over this cycle, and this is, remember, is a simplified version of Kelvin cycle. After this we will see the more complicated and the detailed version of Kelvin cycle, but before that we need to understand this. Carbon dioxide from the atmosphere is accepted by RUBP, rubulose biphosphate. We have to keep a track of carbon so that we are able to trace everything, and all these molecules or their names which we are writing, they have been given to us by Kelvin, by the experiment they performed, in which they kept killing chlorella cells at regular intervals and analyzing which products are formed. And this is how we know that the first product is this, then this, and so on. So RUBP, which is a 5 carbon compound, accepts 1 carbon dioxide. To make this understandable, we are starting with 6 molecules of CO2 accepted by 6 RUBP. Enzyme which is going to help is Rubisco. The special things about Rubisco, it is the most abundant protein on the earth, and its behavior changes in certain conditions. Normally it acts as carboxylase, but in certain conditions it is going to act as oxygenase. We will see that later on. So Rubisco helps in addition of carbon dioxide. That is step number 1, carboxylation. The first stable compound which is formed is a 3 carbon compound called phosphoglyceric acid. There is a short lived temporary compound also formed which scientists are not very sure of, but they think that in first is a 6 carbon compound. As soon as it is formed, it is dissociated. They are not able to trace it or extract it. That's why we just simply write it and then first stable compound. The first stable compound is a 3 carbon compound, and that is why Kelvin cycle is also known as C3 3 cycle. First stable compound, phosphoglyceric acid. This phosphoglyceric acid changes from one phosphorus containing molecule to or compound to 2 phosphorus containing compound. So instead of phosphoglyceric acid, it becomes 1,3-diphosphoglyceric acid. Phosphorus and energy is required, that comes from ATP. 12 PGA are changing into 12 1,3-diphosphoglyceric acids. So 12 ATPs will be used up here. Now this diphosphoglyceric acid undergoes reduction. It gets reduced into aldehyde and NADPH2 gets oxidized into NADP. So this reducing power is utilized. 12 molecules of 1,3-diphosphoglyceric acid get reduced. So 12 reducing agents are required and we get phosphoglyceraldehyde. Phosphoglyceraldehyde is one phosphorus containing compound, and that is why 12 inorganic phosphates are given out, because it is a diphosphate and this is single phosphate one. 12 molecules of phosphoglyceraldehyde are formed, out of which 2 get separated from this cycle. When 2 molecules are separating, that means 6 carbons are getting separated, and these 6 carbons will give us glucose. Again, this glucose is not going to synthesize like a straight glucose. It is first fructose, then glucose. That we will see when we come to that complicated actual version of Kelvin cycle. So out of 12, 2 get separated to synthesize glucose, and 10 will help in regeneration of RUBP. So these 10 molecules means 30 carbons. RUBP started with 6, each 5 carbon containing, so 30 carbons. RUBP is a biphosphate and phosphoglyceraldehyde has only one phosphorus. So for 6 RUBPs we need 6 more phosphorus or phosphates. So they are coming from 6 ATPs and RUBP gets regenerated. So this complete cycle can be explained in 3 steps. First is carboxylation, then is reduction of phosphoglyceric acid into phosphoglyceraldehyde, which takes place in 2 steps. It is similar to the reverse of glycolysis, a part of glycolysis where glyceraldehyde changes into phosphoglyceric acid. So it is same as reversal of glycolysis. And the third step is the phosphoglyceraldehyde molecules help in regeneration of RUBP. So this RUBP is again available for more carbon dioxides to come in so that it can accept it. Rubisco, very important enzyme. All other enzymes are like everywhere ATP is there, it is called phosphorylase. It will be called a reductase. So here the enzyme is a reductase. Here it is again a phosphorylase. This is a simplified version of Kelvin cycle. The complicated or the actual Kelvin cycle is not like one step. There are multiple options which are possible for certain molecules in the plant. So next is the actual version of Kelvin cycle, but before that, dark reaction. The site of this dark reaction is stroma. Site of dark reaction is stroma. Light reaction takes place in thylakoids because the pigments are there. So dark reaction in stroma. It is known as Kelvin cycle, who actually gave us the path of the complete cycle, traced all the intermediate compounds which are synthesized, and another name is C3 cycle because the first stable compound which is formed is a 3-carbon compound. So now let us talk about the complicated one or the actual Kelvin cycle.